Melissa, thank you very much for a very profound and very well put together presentation, which shows that our regulators are open for business. And this is certainly a very admirable response to the challenges that we face. I'd like to call on our second speaker, who is Dr. Kumiko Sakai Kato from the National Institute of Health Sciences from the Ministry of Health, Labour and Welfare from Tokyo in Japan. Please, Dr. Sakai Kato, the floor is yours.
Thank you very much for introduction, and also thank you very much for inviting me to these meetings. I'm very glad to be here again. So I'd like to update the activities of regulatory science research for nanomedicines in Japan. And disclaimer, these views and opinions expressed in this presentation are those of the presenters and do not necessarily represent official policy or positions of the Ministry of Health, Labour and Welfare. So the research into the applications of nanotechnology to pharmaceuticals is very active in Japan. Government researchers at NIHS MHLW are keeping up with the latest relevant scientific findings and promotes the regulatory science researches. And especially the analytical method of nanomedicines and the evaluation method of biodistributions, including intracellular trophic nanomedicines, is those of the examples we are working on. And open discussion would be followed between industry, academia, and regulatory authorities about the appropriate regulations of nanomedicines for enhancing the medical application of this technology. And international cooperation with other organizations through IPRF is also important. So based on these findings, research outcomes, and dialogue with industry and academia, MHLW is developing points to consider documents for development and manufacturing of nanomedicines and exploring the possibility of the future regulations of nanomedicines. So for innovative drugs like nanomedicines, points to consider for requirements of first in human study is also important for ensuring the quality and safety. And these are guidelines or reflection papers specific to nanomedicines recently published from the Japanese Ministry of Health, Labour and Welfare. The guidelines for the development of liposome drug products, reflection paper on nucleic acids, siRNA-loaded nanotechnology-based drug products. These two documents have been published this March. And MHWMA reflection paper of block polymomyces mesna product is published in 2014. I will introduce the two new guidelines. This is a table of content of guidelines for the development of liposome drug products. This document mainly provides information regarding the pharmaceutical development and non-clinical and early clinical studies of liposome drug products. So this document contains the points to consider in development, the so-called active targeting liposome using the ligand or antibody for the active targeting to some cells or tissues. So it also contains the points to consider for a compatibility assessment of liposome drug products subject to change in their manufacturing. So you can get the document from the website of Division of Drugs of National Institute of Health Sciences. And novel nucleic acid products are actively developed, and the efficient delivery system is one of the essential tools for the successful clinical application of some oligonucleotide therapeutics such as siRNA. This reflection paper discusses some of points for considerations when assessing siRNA drugs product made using non-technology-based carriers. This is a table of content of the reflection papers, and this document discusses the quality, non-clinical, and the first-in-human study considerations, especially in terms of pharmacokinetic behavior, delivery to target cells, and safety of the drug product. So regarding to the carrier-specific considerations, the relevant notifications and guidelines, such as liposome guidelines or block polymyxin reflection papers, should be consulted. So I added one slide to show one of the examples of research on the analytical method of nanomedicines using the atomic force microscopy. So this is, I'd like to introduce one of the regulatory science research at NIHS. Although it is still basic research, we recently developed an experimental techniques that allows a systematic analysis of liposomes in an aqueous medium by using AFM, regardless of variations in membrane surface charge, phase state, or degree of saturations of the lipid acid change, and PEG modification status. So mechanical rigidity of the liposome membrane is often defined by the mechanical rigidity of the liposome membrane is often defined by the membrane bending modulus, but the quantitative experimental data are still limited to a few kinds of liposome because they easily collapse and form a supported lipid bilayer on the solid substrate. So our established AFM system allows to investigate the membrane bending modulus of nano-sized liposomes in aqueous medium. This graph show below. Graph show the bending modulus of liposome with various lipids. So as shown here, the bending modulus decrease the foreign orders, solid ordered, liquid disordered. So this data show that the bending modulus obtained by means of AFM could be parameters, quantitative parameters of membrane rigidity. So this is, we are continuing the research to obtain the validation of the established methods. So this is one example we are working on at NIHS. So thank you very much. This is the update from the Japanese NIHS. Thank you for your attention.